Good evening, everyone. I hope I'm audible to all. Welcome to this session on developing and writing protocols. Let me quickly share the screen so that you all can see my presentation, and then we can start from there. Okay. Okay, welcome once again to the session on developing and writing protocols. This session is brought to you by Walters Kluwer and Cactus. I am Vihang, your host for the session today. Let's first understand what is Walters Kluwer. This is an organization that is already serving 180 plus customers, serving several customers in 180 plus countries, operations in 40 plus countries. They have approximately 20,500 employees all over the world, and there is a euro 5.6 billion euros of revenue, and they do serve 93% of Fortune 500 companies. The values that they share are focus on customer success, making things better, aim and high and deliver, and win as a team. A little bit about Cactus. Cactus is a company that has been serving us for the last 20 years, specifically providing support to the authors, researchers, and publication companies to publish and write their science better. It's more than 23 years of success, 1,300 plus people who are working with these organizations, and 5,000 plus scientific collaborations worldwide, and they have offices in 7 different countries. Cactus has 4 major products out there in their portfolio. One is Editage, which actually has 20 plus expert services and AI tools to meet research and publication goals. Then there is PaperPal, which is a complete AI writing toolkit for academics. Then there is R Discovery, which is the #1 AI platform for literature search and reading. And finally, there is Mind the Graph, which is a service for making scientific figures, graphical abstracts, and infographics. So that's all about the organizers of this session. A little bit about myself. I am Vihang Galsasi. I hold a PhD in molecular biology from Heidelberg University, Germany, and a master's in biotechnology from IIT Bombay. I have previously worked as an assistant professor at Shulini University in Himachal Pradesh, and I have worked in industry for over 6 years. I have a mix of experience in academia as well as industry and entrepreneurship, and I've been working or been associated with Cactus for more than 5 years as a freelance copy editor and as a trainer. Until now, I have helped several researchers in publishing their work through copy editing and consultation. So in today's class, we are going to talk about how to write and develop protocols specifically to clinical research. But before we start, let's understand why writing is important. See, as a scientist or as a researcher, you might have had your eureka moment. You might have discovered something new. You might have developed something breathtaking. But if you don't tell this to the world, how will even the world know that you have done something great? How will they even use that particular innovation or that discovery for the benefit of society? So in this case, writing and communication become super critical. If you don't do it well, it's as good as you have not done your research well, right? So just to make that research successful, writing and communication is an integral or a critical part. More to that, it is important to ensure data accuracy and reproducibility. It is important to avoid data misinterpretation. It is important to maintain research integrity, to ensure compliance to ethical guidelines, to meet regulatory standards, to facilitate drug development and commercialization, to ensure patient safety, to protect business interests, and finally to benefit society, right? So that's the reason why writing is important, and all of us, whoever is engaged in research or innovation or anything scientific, should definitely learn writing well so that we don't miss out on being successful in research in the long run. So let's talk a little bit about structure of a typical research article. Many of you who are students or research scholars must have read a lot of scientific literature by now. And you must have noticed that every research article more or less has a format called IMRAD format. What does it stand for? It stands for introduction, methods, results, discussion, conclusion, and finally an abstract. So introduction states context, purpose, and hypothesis. Methods generally describes materials, protocols, and analysis. Results presents and analyzes data. Discussion explains whether the hypothesis is proven right or wrong, and it also talks about limitations of the study. Finally, conclusion summarizes the key findings and states prospects. An abstract, as you know, It is a concise overview of the entire study. So because this session is more about clinical trials or clinical research, let's understand what are clinical trials. Okay, clinical trials are actually research studies on humans. Say when a new medicine or a new drug is coming into practice. Just a few years ago, we had COVID pandemic, and during COVID pandemic, the scientists came up with a vaccine. Now that vaccine, when before it was introduced to the patients or to the people, it had to go through several rigorous studies, and those rigorous studies are actually clinical trials. These are research studies with human volunteers to evaluate interventions. Now, what's the purpose of this evaluation? It is to determine if new treatments are safe, effective, and potentially better, right? If you developed a drug, but how do you even tell the world that it is better than existing drugs, or it is a new drug that can sort of cure a disease that was earlier not curable? Correct. So from that point of view, clinical trials are important to evaluate a particular new treatment regimen. It contains systematic phases. The progress has by and large, they are divided into 4 phases, 1, 2, 3, and 4, and every phase has a specific objective associated with it. The clinical trials are strictly regulated. They have a very rigorous oversight. They require approval from health authorities and from ethics committees. Evidence of approval means data is essential for regulatory approval of new interventions. So this is what exactly the clinical trial aims for. It aims for generating data so that it can be given to regulatory approval and that the drug can be eventually brought into the market. Advancing medical knowledge, yes, the whole activity, even if some of the trials may fail, they don't work out, they say that the drug is not effective. Even in that case, the whole experiment or the whole activity is important to expand our knowledge in understanding disease and contribute to future research. So let's understand what is the difference between a original research article, a generic original research article, and a clinical research article. An original research article and clinical research article, we can just call them as OR and CR for our convenience right now. Both OR and CR follow the IMRAD format, correct? In introduction, methods, results, and discussion. The nature of hypothesis or the scope for the original article is often exploratory. It encompasses diverse scientific areas. It could be basic science, mechanisms, novel discoveries. It can be plant biology. It can be geology. It can be, it can be molecular biology. It can be anything. But for clinical research, it is specifically about evaluating specific interventions, which might be either drugs, devices, therapies, or diagnostics in human subjects, right? So clinical research is specifically in human subjects. The method section in original research includes material, sample collection, experimental procedures, methods, instrumentation details, and statistical methods. So here the method section is sort of a generic, and it also changes as per the actual question in mind. So if it is plant biology, it will talk about where the plant sample is collected from, how it is processed, how it is stored. If it is a molecular biology, it will talk about DNA isolation or other DNA RNA related techniques. But in clinical research, it is always about study objectives, patient selection criteria, intervention details, data collection, monitoring, and statistical methods. So clinical research, because it caters to clinical evaluation, it has a certain defined set of sections within the methods section of the article. Ethical considerations, in case of OR, they may or may not be relevant, because as I told, if OR is related to any non-living entity, say you are studying rocks, or you are studying some soil sample, or you are studying some other entity, then ethical considerations may not be relevant. But if an OR involves human samples indirectly, it might not be clinical in nature, but if it's say you're taking blood samples from people or you're taking tissue samples from animals, then yes, in that case, original research also involves ethical considerations. Clinical research, on the contrary, always includes a prominent ethics section detailing institutional review bodies or ethics committee approvals and informed consent, right? So ethical considerations in the case of clinical research are very important, and they form a prominent part in the methods section of the CR. We're going to talk more about it in upcoming slides. Guidelines and templates. OR follows basic scientific principles, and there is less emphasis on any rigid templates. But in the case of CR, there are very strict protocol templates and guidelines such as ICH, GCP, Consort, etc. We are going to talk about these as well on the upcoming slides. So let's go through each and every section of a manuscript, of a clinical research manuscript, and understand what's there in all those sections, and then finally go on to the methods section, wherein the protocols are written and understand more about guidelines, understand more about what needs to be written in that section, and go deep diving into this particular topic. The introduction section, it contains background and rationale of the study. It talks about significance and importance of the study. Unmet medical need, which means what? That, oh, there is a certain disease, there are so many people being affected because of this, and they're having a poor quality of life, and there is really nothing much that can help these patients, or whatever the therapies or medicines that are available, they are not sufficient, or they are not giving you satisfactory results. That is why there is a need to make something new or to change something in that. This is what you need to mention in the introduction section. And when you highlight the unmet medical need, you also give a brief overview of existing literature. You tell them that, look, all of this has been done until now, but there is still an unmet need, and this is what my hypothesis is about. This is what my hypothesis or objective is about, right? So this is what you write about in introduction. Next is about publication section of methods. We're going to go more detail into it, but this section involves what? The study design, patient selection, interventions, data collection and monitoring, statistical analysis, and ethical considerations. As I already talked about in the previous slide, that ethical consideration becomes an important section in the methods section of a CR. Then results talk about patient enrollment, baseline assessment, data collection and monitoring of different parameters, data presentation in the form of images, charts, and tables, and then analysis of this data using relevant statistical techniques, right? So whatever things that you have observed during the entire course of your clinical research, what was the blood pressure of all the patients, how much was their weight? Just excuse me. I'm sorry for the interruption. Okay. Yeah, I'm back. So the results section contains patient enrollment. That means how many patients were actually involved. What is their baseline assessment? What was their actual health status in the beginning of the clinical research? Then it talks about data collection and monitoring, like maybe their blood pressure was assessed, or maybe their body weight was assessed, or maybe their heart rate was assessed. There could be multiple such parameters. Then finally, it contains data presentation in the form of images, charts, and tables, like, you know, how in the cohort, how many patients showed what kind of a change throughout the course of the trial. And then the analysis using relevant statistical techniques. So what stats have you used? What kind of a tools have you used to compare different groups? So on and so forth. The next section is about discussion. It's about data interpretation in the context of objective and hypothesis. Data comparison with prior literature, novel outcomes, stating what new have you observed over here. Then it's about strengths and limitations of the study and path forward. See, no research is perfect. It will always have some limitations. Either the size of the patient or the patient pool is not large enough to make conclusions, or it is about some data points are missing, or it is about some regimens were not implemented properly. It could be anything, correct? And because it involves humans, there are always some unexpected outcomes. Patients drop out of clinical trials sometimes, right? So multiple such things happen, and then those things contribute to limitations of the study, and then these need to be explicitly stated in the discussion section. And the whole outcomes that you have achieved, they need to be compared with prior literature, and you need to make a commentary on what is new that you have observed in the entire study. And finally, you need to talk about path forward, which means what next. So you will say that, yes, my treatment was effective, but it needs, you know, some more amount of study on some other parameters to actually take it to the next level. So that's all about the discussion section. So now let's dive deep into the protocol section, or the method section, where you are going to write about the protocol in detail. Now, what is a clinical trial protocol? It's a detailed written document that describes the objectives, design, methodology, statistical considerations, and organization of a clinical trial. Correct? So understand these words very well. A clinical trial protocol describes the objectives, design, methodology, statistical considerations, and organization of a clinical trial. It is essential for ethical approval, regulatory submission, and study execution. Ensures consistency and standardization across sites. Now, let me highlight a key difference over here between the OR and CR. In original research, what happens? Your method section evolves over time. right? You have some objective in mind, you do some experiments, you see that, oh, maybe this is not really matching. Then you make some changes, finally you refine your parameters, change the experiments, and then eventually whatever outcome is found, or whatever the key results that you have found in your manuscript, you only write those methods that have actually contributed to that outcome. Correct? You don't write about failed experiments, or you don't write about experiments that were inconclusive. But here, and you don't necessarily have to formulate the entire methodology and seek anybody's approval in the case of OR. But in the case of CR, because there are ethical considerations involved, you need to develop the entire protocol beforehand. You need to identify what is the expected patient size, what is the expected patient population, what is the expected exclusion criteria, what are the primary and secondary endpoints, everything, the whole methodology, the whole design, and submit it for ethical approval, submit it for regulatory approval, get their actual approval, and then start your study. Correct? So again, that doesn't mean that you cannot change anything later, but if you want to adapt anything, there are also adaptive trials, by the way. We'll talk about it later. But in those cases, again, every step you change something, you still need approvals. Correct? So that is the reason why this entire session is named as developing and writing protocols, and not just writing protocols. Right? I hope I am able to make this distinction very clear. In the case of clinical research, you need to develop a clinical trial protocol first, seek regulatory approval, seek ethical approval, and then execute the protocol. And in case if your trial is adaptive in nature, every time you make changes, you need to have a rigorous validation of those changes through regulatory and ethical bodies. So what are the guidelines that are present for protocol development? There are these 5 important ones: NIH protocol templates, SPIRIT, which stands for Standard Protocol Items Recommendations for Interventional Trials, CONSORT, which means Consolidated Standards of Reporting Trials, CARE, which means Case Reports, and ICS-GCP, which means International Council for Harmonization, Good Clinical Practice. Right? I'm not going to read through all of this, but I'm going to explain each and every template to you in short. What does NIH protocol template have? The focus of this is to provide specific protocol templates to assist applicants in writing clinical protocols for various types of research. Right? So there are like templates. So templates matlab kya, like how you fill forms for things, right? You have everything that is already mentioned, and you simply have to fill in some fields. So likewise, there are templates that are available for phase 2 or 3 clinical trials requiring investigational new drug or investigational device exemption applications, and for behavioral and social sciences research involving humans. So multiple different types are there, and for those there are templates available. The purpose of doing this is what? To offer instructional and sample text aiming to facilitate comprehensive protocol development that meets standards like ICH Good Clinical Practice, and to streamline review by oversight entities. Correct? Now understand why all of this is important. You might be conducting your study in India, you might be conducting in Japan, you might be conducting it in Brazil, or maybe in Mexico, or anywhere in the world. Like take an example of COVID vaccine. So all the clinical trials that were conducted, they were conducted in multiple parts of the world. But eventually the vaccine was given to all the citizens of the globe, right? So some kind of a standardization is required, so that a trial that is conducted in one population, in one particular geography, can be understood by other geographies, and their own laws and legal regulations and regulatory standards could be adapted based on that. Right? The whole commercialization can happen in other countries based on the results that you have obtained in one country. So for this to happen smoothly and seamlessly, you need to have rigid guidelines and rigid protocols, which will make everything easier for regulatory authorities to assess. So accessibility protocols can be developed using a secure web-based e-protocol writing tool, or by downloading Word versions of these templates. Correct? So there are again a lot of softwares wherein you can use some tools to fill in these templates and develop everything correctly. Relevance is to you. This use promotes a common protocol structure and organization essential for consistency and regulatory compliance in NIH-funded clinical trials. Now, NIH is a very large organization, and it funds clinical trials all across the globe. So to ensure consistency across all those organizations, this kind of a protocol structure is very necessary. Now, what is SPIRIT and CONSORT? Here, the focus is about a globally recognized checklist detailing the minimum content items that should be addressed as in a clinical trial protocol. Now here the NIH is literally like a template of complete protocol, but here it's just like a checklist. It enhances quality, completeness, and transparency of clinical trial protocols, facilitating better understanding and evaluation by stakeholders like ethics committees, funders, and researchers. The structure here is what? It is organized into 33 items covering administrative information, introduction, methods, study design, participants, sample data, sample collection, statistical methods, and ethics and dissemination. What is the benefit? It promotes consistency in protocol content, reducing heterogeneity and improving the comparability of trials. Again, the same example that I gave you about COVID, correct? So trials are being conducted somewhere around the globe, and then the results are actually used everywhere in the world to commercialize that particular vaccine or a drug or a medical device. So this is where, to ensure consistency, to reduce heterogeneity, these kind of checklists are important. Next is care guidelines. So care corresponds to case reports. It provides framework for the accurate and transparent reporting of individual case reports, enhances the utility and value of case reports by ensuring essential information is included, right? So this is to identify, early identification of new diseases, adverse drug reactions, or unusual clinical presentations. It consists of a 13-item checklist and flow diagram covering details like patient information, clinical findings, timeline, diagnostic methods, therapeutic interventions, and outcomes. The relevance here is particularly useful for initial safety observations in early drug development phases or reporting rare events not typically captured in longer trials. So the care guidelines here are specifically about more relevant about say phase 1 and 2, where you are having a preliminary observation of any adverse event. So you have a group of say 10 or 15 people, and in one particular patient, you observed some unexpected outcome, some unexpected reaction or something. How do you actually document that? So there is a 13-item checklist for it, correct? There is a proper diagram for documenting that information. And then these case reports have to be actually included in the whole data recording database of the clinical trial, so that you make necessary changes or seek the relevant ethical approvals. Last is ICH GCP. So this is an international ethical and scientific quality standard for designing, conducting, recording, and reporting trials that involve the participation of human subjects. So these are GCP practices, good clinical practices. The purpose is to provide a unified standard to facilitate the mutual acceptance of clinical data by regulatory authorities in different countries. The key principles here emphasizes protection of human rights, safety, and well-being and trial participants, adherence to the protocol, quality assurance, and proper documentation. The scope covers responsibilities of various parties, including sponsors, investigators, and institutional review boards or ethics committees. So finally, after looking at all the guidelines, let's understand the core sections of a clinical trial protocol. First is rationale, then objective, study design, study population, intervention methodology, data collection and monitoring, statistical methods, and ethical consideration. Now, what is the rationale? Correct? Rationale means, as I said, what are you... Now, don't get confused between what you write in the introduction and what you write in the rationale section of the method. Correct? In the introduction, it is like the larger long-term goal that you want to say. You can say, oh, I want to cure a particular type of breast cancer, or I want to conduct a trial to understand if a medicine works against a particular cancer. That's like your long-term larger objective that you have already stated in the introduction. But here in the method section, you're talking about very specific objectives. Correct? The main question that the trial aims to answer. It talks about clinical endpoints and what is exactly the hypothesis that you want to talk about. So these objectives have to be SMART. They need to be specific, measurable, achievable, relevant, and time-bound. Next section is study design section. This is, I think, the most important section of the entire clinical trial. It contains, generally, clinical trials are of 4 phases. So depending on your trial is of which phase, you need to explain that in this particular section of the methods section, right? So either is it phase 1, is it a first in human studies, which is a small focused on safety, dosage, and pharmacokinetics? Is it phase 2, which explores efficacy and further evaluates safety in a larger group? Is it phase 3, which is a large-scale pivotal trial to confirm efficacy and monitor adverse event? Or is it phase 4, which is about post-marketing studies to gather additional information? Whatever your study is, your clinical trial is, in the study design section of the method section, you need to emphasize which phase it belongs to. Next is about what type of a trial it is, like what phase you have already mentioned. Now, what type is it? Is it an RCT or is it an observational study? Is it prospective? Is it retrospective? Is it placebo controlled? Or is it, what kind of a grouping has been done, right? All these things you need to mention. Now, I'm not going to explain you the definitions of all of these, but please remember these keywords. Is it a randomized clinical trial? Is it an observational study which is prospective in nature? Or is it an observational study which is retrospective in nature? Is it the gold standard, which is generally considered, which is placebo-controlled trial? What kind of a blinding is there in these trials, right? Is it single blind, double blind, or triple blind? Now, what does single blind mean? It means either the participant or the research team is unaware of the treatment assignment, which means whoever are the trial participants, they don't know whether they are given placebo or the drug, right? But the people who are administering drugs, they know everything. And even though people who are keeping the oversight, they also know everything. Double blind means both the participants and the research team are unaware of who is receiving what experimental outcome versus the control. That means if I am a doctor or I am one of the healthcare professional who is conducting the trial, even I don't know which patient is getting what. And even those patients, they also don't know whether they are getting placebo or whether they are getting actually the treatment regimen. In triple blind, participants, researchers, and data analysts are all unaware of the treatment assignments until the study is complete. So somebody out there who is like at a senior position in senior healthcare professional designs everything, allots that this is a group of patient who gets this is the patient of group that groups gets like a placebo, and executes everything. But in the entire process, neither the patient nor the people who are administering drugs, nor the analysts who are doing statistical analysis, any of them are actually not aware of who gets what. So this is a very higher level of administration where you avoid any kind of an individual bias. Lastly, is it a placebo-controlled trial? I think I talked about it on the previous slide. And then there are other specialized designs, something like adaptive trials. I told you, right? Like a trial design can be modified based on accumulating data, which is sample size re-estimation, arm dropping, etc. So such trials could be some specialized designs. Then there is a non-inferiority, superiority, or equivalence trials, which is designed to show if a new treatment is not worse than, better than, or as good as an existing one, right? And then there is also something called as N-of-1 trials, where is a single patient is involved in a trial. This is particularly relevant for rare diseases or genetic disorders, which are, found maybe only 1 in a million or 1 in 10 million. So you are definitely not going to get a lot of patients to conduct such trials. So such trials are generally conducted on a single patient. The crux of this entire thing is that you need to correctly mention what is the design that you have in your given trial. Now, all these types are not mutually exclusive. It doesn't mean that something is a randomized controlled trial. That means it cannot have a specific blinding, or it can have only this type of a blinding. Multiple overlaps can be possible in defining or describing the design, correct? The point being, in the study design section, you need to clearly mention what is the kind of this particular research, clinical research that you are conducting. The next section is about study population. Determining patient size. This is calculated based on primary endpoint, expected effect size, statistical power, and significance level. Ensures sufficient power to detect a clinically meaningful difference, right? So a particular disease, how much is the overall incidence of the disease in the population? That means how many patients I really need to test my drug on to statistically significantly conclude that my drug is effective. So there is a lot of principles and a lot of statistics involved in determining this, but that needs to be done, and then accordingly you need to justify that why this much of patient size is important, or this much patient size I have selected. So this justification of sample size is critical for ethical and scientific validity, and over or underpowering can lead to ethical concerns or inconclusive results. Say a particular drug requires that you need to have 10,000 people to conclude something significantly, but you do it only on 1,000 people. That means you are underpowering the trial. On the contrary, it requires only 5,000 people, but you're doing it on 50,000 people. That means you're overpowering the trial. And in either case, there are ethical concerns involved, and it could also lead to inconclusive results. Defining patient population, correct? You need to have a set of criteria to define what should be their age. Should they be? Should it be men or women? Should it be of? Should they be smoking or non-smoking? What should be their weight? What should be their lifestyle? Correct. So all these things have to be there. Should they be having any specific disease beforehand? Correct. So all these things are involved, and accordingly you have to decide whom to include and whom to exclude. This is crucial for defining the target population and ensuring patient safety. Next is intervention methodology. This is a precise description of the investigational product or intervention. What is the exact molecule that you are administering? What is its dosage? Dosage means how many milligrams or how many micrograms in and how many times in a day, for how much duration, for a week, for a month, for 6 months, or whatever. Route of this administration, is it intravenous, intramuscular, is it oral, or is it intraperitoneal? What is the route of administration? Frequency, how many times in a day, and duration means for how long, right? Blinding procedures, I think we talked about single, double, and triple blinding in our previous slide. So that needs to be mentioned in the intervention methodology as well. Rescue medications or concomitant therapies, allowed or disallowed, correct? That means if some adverse events are there, then are there anything that you can use to rescue the particular patient, or to just to reduce the effect of the current drug? Is there anything else that is being used? Handling and storage of study materials, how it is done, correct? So in case if you are taking any tissue samples for analysis, are there any biopsies conducted, or are there any blood samples collected? Then how are you collecting them and how are you storing them? Data collection and monitoring, correct? So here case report forms, the one that we discussed in our previous slide, is important. Source documents, like that is medical records and lab reports, schedule of assessments, training for study staff on data collection procedures, and quality control measures for data accuracy. All these things are important for data collection and monitoring. Finally, statistical methods. It's a detailed plan for how the data will be analyzed to address study objectives. Choice of statistical tests based on data type and study design. Handling of missing data, interim analysis is required. Definition of analysis populations. A separate comprehensive document often developed alongside the protocol. This is very, very crucial. The statistics part in clinical research is quite complex. It is very well articulated. There are books on it. There are several guidances on it. So this also needs to be developed. This also needs to be properly formulated before you present your developed protocol to your ethical and regulatory authorities. Finally, the ethical considerations. So informed consent process. Informed consent is the super critical, most important thing. Every patient, every individual who is participating in the clinical trial should be informed about what kind of a thing that they are going to go through. It should be clear, comprehensive, and voluntary. You cannot force anybody into a clinical trial. You cannot give them any misleading information. You cannot scam them. You have to clearly convey them that, look, these are the risks associated with this particular trial that you are going to go through. Are you aware of all of this? Are you really aware fully about what you are committing to? Is it your voluntary submission, or is somebody forcing you into this? So all these things are very, very important, and they need to be submitted to the institutional review board and ethics committee approval. Patient privacy and confidentiality. Now there are separate guidelines for that. HIPAA and GDPR, these are like separate guidelines for that. The personal information of patients has to be maintained confidential, correct? You must have seen in news COVID clinical trial, this is the efficacy, this is the safety, etc., etc. Did you anywhere see who were the people who participated in the clinical trial? No, because this is a very clear guideline that the patient personal data, their name, their affiliations, their their family members' names, or all of that stuff has to be kept fully confidential. Risk benefit assessment for participants, data and safety monitoring board, oversight for certain trials. So do you have like a separate board who is going to oversee the entire safety and all that stuff? All these things are very important in ethical considerations. So what are the best practices for writing protocols? First is start early and allocate sufficient time. Second, collaborate with multidisciplinary experts, clinicians, statisticians, and regulatory. Now it's a multidisciplinary affair. The person who is going to check the pulse or record blood pressure is probably doesn't have any clue about what statistics is there. The person who is going to run all these T tests and Z tests and whatever, all sorts of tests on the data, doesn't know how to actually take a needle and inject it into a person, right? So there are different expertise which are very specific, and you need a multidisciplinary collaboration for developing the protocol. Use templates and checklists. We talked a lot about templates and checklists earlier, so you should be using them while writing such protocols. Be clear, concise, and unambiguous. Version control is crucial. What is version control, by the way? You wrote a draft right now. Then maybe a day later, somebody gave you suggestions and then you revised it and another version is created. Then you submit it to authorities. Authorities told you, no, these are points that are missing. You need to make these changes. And then you made those changes and then another version is made. So all these versions have to be clearly labeled. These files have to be clearly named. They should be sorted in different folders and all those has to be very clearly controlled. Peer review and expert feedback are invaluable, correct? So you need to ask feedback from experts. You need to ask feedback from your peers and from regulatory authorities to ensure that your protocol is very well adapted for the purpose that you are investigating. What are the common footfalls in protocol writing? Vague objectives or endpoints, correct? Your clinical endpoints have to be very, very clear, crisp, and precise. Inadequate sample size, correct? I explained this a little bit earlier that based on the incidence of the disease, you need to exactly identify what should be the size of your patient population to conclusively come up with results, right? So this kind of a sample size is important. If you have an inadequate sample size, then you are underpowering your clinical trial. Unrealistic timelines, correct? This is after all biology. This is something that involves human patients. So you need to consider what is realistically possible timeline for executing this entire trial and only commit to that. Poorly defined inclusion-exclusion criteria. Yes, you need to define your inclusion-exclusion criteria properly to ensure that you are not, introducing any biases in your entire analysis. Lack of clear intervention details. Now, intervention details are what? What kind of a drug was administered? What was the dosage? What was the frequency, duration, route of administration? What are the, what are the parameters that are being observed? All of this has to be very clearly recorded. Insufficient statistical planning. Again, take help from experts, take help from statistics, the people who are experts in statistics, and ensure that the correct kind of tests are conducted on the data. Overlooking ethical considerations. This is a very common pitfall. Ethical considerations can be, the whole set of guidelines could be sometimes very intimidating. It is very detailed. So it sometimes happens that you oversee something or you don't happen to take things to the notice very clearly. And then that might end up in your protocol being rejected. So protocol development is an iterative process. First is drafting, review, and feedback and revision cycle. Next is input from various stakeholders, such as your sponsor, investigators, statisticians, and regulatory. And third is protocol amendments may be necessary as the trial progresses. Correct? I think I talked about it, that as the trial progresses, there might be some changes that might be involved, and you need to again go through a process of development, writing, and approval. So what are the key takeaways from this particular protocol writing session? Protocols are the cornerstone of high quality clinical research. Adherence to guidelines such as SPERIT, ICH, GCP, or CONSORT or PubMed templates is very essential. It should have clear objectives, endpoints, and robust methodology. These are all paramount. Collaboration and meticulous attention to detail are critical for success. Now, before I move on to the next part, I would like to take a small pause if there are any questions, because I have been just talking for a long time. Please feel free to drop your questions in the Q&A. Okay. Lokeshwar has asked, is it always necessary to state the hypothesis or research question even when we state the objectives? Right. So Lokeshwar, I think I made it clear that your introduction has a larger goal and the objective of your clinical trial design is a more specific goal, because it actually emphasizes on the clinical endpoints that you want to achieve. So yes, both of them are necessary. In the introduction, you're going to tell that I want to cure a certain type of cancer. And in the actual objectives or rationale section of your protocol development, you're going to say that I want to achieve this clinical endpoint. Correct? So both of these are very different. I hope I answered your question. Are there any other questions? I'll wait for another 30 seconds or something. Please feel free to drop these questions here in the Q&A chat bot, and then I can take it up later after I finish the entire session. Okay, so looks like there is nothing else. I'll just continue with the later part now. Okay, I hope my screen is visible. So after we have discussed the entire process of protocol development and writing, after we have discussed ethical considerations and everything, I'm going to focus a little bit on choosing the right journal and going through the process of publishing. Right? So you have done your study, you developed your protocol, you got your approval, you conducted a clinical trial, and now it is the time for you to publish your study. So how do generally authors choose a journal while publishing? First, they look at is the scope of the journal. Correct? Scope means what does the journal want to publish? Correct? You have done a cancer research and you can't submit it to malaria research. Correct? I'm giving a very vague example, but the scope of the journal is important. Publication metrics such as impact factor, site score, indexing status. I believe all of you know about these things. Timeline of publishing. How much time a journal takes to publish something. Stringency of peer review, right? So how rigorous or how meticulous is their peer review. Next is whether it is open access or subscription based, and if there are any article processing charges. So I'm going to talk a little bit more about open access and other subscription models or other publishing models on upcoming slides. So there are different publishing models. First is called as an open access model. This is something where research is made freely available for all to access. Correct? Removes financial barriers for readers, charges the author or the institution or the funder via an article processing charge. Correct? So understand one thing, for any publishing house or for any journal, there has to be some source of income for them to maintain their operations. Correct? So because they screen for your articles, then they select the one that is fit for their purpose. They conduct peer review. They maintain an online presence. Correct? They promote your article after publication. There is typesetting, there is copy editing, there is proofreading. There are a lot of things that are involved. All of these things you need to hire people and you need to maintain an organization to conduct all these things. So obviously from somewhere the money has to come in, and either that money will come via subscription. That means you either charge the reader or you charge the author. Correct? So in open access, what happens? The reader doesn't have to pay. Readers can just go click on the article and read it freely without any barrier, but the author has to pay because obviously the publication house has to maintain their operations and maintain their source of income. Next is a subscription based model. Yahan pe what happens is access to research requires payment, which means the reader has to pay to read the article. I believe all of you are browsing enough amount of literature online, and sometimes you see that only the abstract is visible and the rest of the article you can't really access because you need to pay for reading that particular paper. So here, the reader or the institution or the library is being charged to read or access that particular paper. Then there is something called as hybrid open access, which is available post acceptance. 300 plus journals are there since February 2013. Author or funder pays article processing charge, and then there are some kind of licenses involved, which is CC BY, NC ND license or CC BY license, wherein if you are using a particular piece of that particular paper by citing its source clearly and without making any modifications, you are allowed to do it freely without any problem. Correct? So this is what is something called as a hybrid open access model. Now within open access also there are two different models. There is something called as gold open access and diamond open access. So gold open access is something what immediate open access, which means final published version of the article is immediately and freely available online. The author pays APC, which is the article processing charge. It's typically paid by the author, their institution or funder. Publisher hosted, which means the content is hosted and managed by the publisher on their journal platform. Then there is another model called as diamond open access, which is again free for all. No fees are charged for either authors or publishing the readers or accessing. It is funded by the community or institution. Correct? So costs are covered by academic institutions, scholarly societies or grants or volunteers. And it's equitable and community driven, often led by the academic community, prioritizing transparency and inclusivity. So diamond open access is something like a middle ground wherein neither the reader pays nor the author pays, but the community pays so that the entire research landscape grows and people benefit from reading each other's research. Now, both OA and subscription models maintain strict peer review, strict and rigorous peer review. Understand this, right? So sometimes people think that open access is there, that means it doesn't have a very strict peer review, but it's not the case. Both have thorough editorial oversight, and both models ensure that consistent scientific quality and integrity is maintained. Now, why OA publishing involves APCs? I think I explained this already, that there is no subscription when it is open access, correct? You are not charging the reader to access the content. And APCs, because they cover rigorous peer review, editing, production, copy editing, typesetting, etc., they need funds for hosting, archiving, and marketing. They need funds for maintaining all of these activities and to sustain the publication infrastructure. That is why in the open access model, there is article processing charge, which is involved. Now, why is open access publishing important? It ensures wider accessibility, greater visibility, correct? I'm sure many of you must have noticed that you are searching literature, you are going and you find a paper that you really want to use for your research, and then you see, oh, it is under a paywall. You are not able to access it. Then you have to go ask some of your friends or ask your institute library to go and download it for you, and then it becomes available to you. So this becomes a barrier for your own research. So what does open access ensure? Open access ensures wider accessibility. It gives you greater visibility. It also protects author rights, correct? Because here in subscription-based model, the copyright actually belongs to the publisher, but in open access, the author retains copyright, correct? So here it also protects author rights. Rapid publication, correct? Because the financial model here is different. Open access is slightly quicker than subscription-based model. And of course, quality, because in either case, there is a rigorous peer review, there is a rigorous editorial oversight. So it definitely also ensures quality research. There are some misconceptions associated about open access publishing. Some people think that OA papers are not peer reviewed. Absolutely not correct. As I already told, open access also involves rigorous peer review. Just like your subscription-based model, open access also goes through the same process. OA publishing is paid publishing. Definitely not true, right? Now, also in the research landscape, there are some malpractices. There are predatory journals which will just come and tell you that, oh, this is open access, you pay us and we'll publish. And they actually do not have any peer review or a rigorous peer review. Such journals are not even indexed on Scopus or on PubMed or on such places. So yes, you have to be very careful in that case in identifying such things. But that doesn't mean that all of open access is paid publishing. Definitely not. If you are targeting a journal or a publisher which is indexed on PubMed or Scopus, which has a good reputation and which is offering you an open access model, then it is definitely a legitimate model of publishing, and it is not paid publishing. OA equals low quality research. Absolutely not true. As I already explained, it goes through rigorous and very stringent peer review process. So it always ensures high quality of research. OA journals are less likely to get cited or indexed. This is also not true. In fact, it is the other way around is true. Because your article is open access, your paper is being read by more people, right? There is no barrier for anybody to read. So the more the number of people reading your paper, the more the chances of your paper being cited, correct? So that way, open access journals are in fact offering larger visibility and better access. OA publishing harms academic reputation. Again, not true, because all of these are misconceptions. You have to be very, very careful about. So what harms academic reputation is actually research misconduct, correct? That is what harms your academic reputation, not open access publishing. Now, what are the roles of stakeholders in open access publishing, right? First is what funding agencies can do. So in the entire research ecosystem, who are the key players? There are funding agencies, right, who give you grants. Then there are universities or institutes who host researchers, right, who give you infrastructure and ecosystem to conduct your research. And finally, there are scientists and researchers like you who actually perform or conduct research-based activities. So what is the role of all these stakeholders? The funding agencies could actually mandate or encourage OA for funded research. This is already happening in Western countries, that a particular grant, if it is given, it says that, look, we are giving you a grant, but you have to ensure that you publish in an open access journal. And it allocates grant money for APCs. So if you get, like, say, for example, 1 crore of a grant, then maybe a part of that particular grant should be reserved for paying the article processing charge. So it directly supports OA model, and it ensures public access to publicly funded research, right? After all, this grant money is public money, correct? So it should be beneficial for the public. Now, what role could universities or research institutes play? They educate researchers on OA. So university is there. University should go and tell all its professors and scientists that, look, OA is good for the society, so you should always publish in OA. Establish institutional repositories, which is called as Green OA. Negotiate, read, and publish agreements. Provide central funds for author APCs. So university could allocate a different set of fund so that researchers or scientists or professors, when they're publishing, they could actually pay for article processing charges. Now, what can scientists and researchers do? They should actively publish in OA journals, right? Advocate for OA within communities, utilize OA resources by driving demand and supply, adhere to OA policies, and manage APC payments, right? So if every one of you who is attending this session goes and talks about OA to their peers, to their friends, to your universities, to your host institutes, automatically there will be more demand for OA, and then the ecosystem will shift towards OA publishing rather than subscription-based publishing. Correct? So this is what the role you as a scientist or researcher could play in making open access more prevalent. I would like to highlight some of the journals from Lippincott or Wolters Kluwer. These are the 5 hallmark journals that they have: Annals of Medicine and Surgery, Medicine, Hepatology Communications, International Journal of Surgery, and Cornea Open. So this is an ophthalmology journal. There's a surgery journal. There is a liver science journal. And apart from these, there are these other journals which are also OA journals, which are open access journals. Correct. So definitely make a note of these, and do consider these journals for your future clinical research publication. Finally, I would like to play a small video on how Lippincott promotes your research. Let's watch it quickly.
Welcome to Lippincott, where we work alongside researchers to make healthcare better every day. We don't just publish your research, we ensure it's seen and valued by a global audience. Our promotions ensure visibility for your research through targeted email marketing and sharing on our active social media platforms. Alongside promotions, we deploy a range of discoverability strategies, ensuring your research is easily found by the research community. All research published with Lippincott is available on Ovid, a trusted platform for over 5 million health researchers worldwide. Our team enhances each article with relevant keywords and metadata to make sure your research appears in relevant searches, both on and off our platform. We also work with a range of discoverability partners, including Web of Science, PubMed, Medline, Scopus, and DOAJ. A trusted partner, we accelerate your research's impact on global healthcare outcomes.
That's it from my end. Thank you so much for your attention, and I'm open for answering your questions. I'll switch off my screen. Okay, there are some more questions. Are review articles accepted from authors who don't have an institutional affiliation? I'm not sure about this, Bimla. Generally, you need to give some kind of an affiliation as to where you are conducting this research from, who is your host institute, etc. Independently, as one individual, if you want to publish, I think you need to check with a specific publisher and understand, right? There is really no one straightforward answer about it. But if you really want to do it, please go ahead and contact a publisher and ask them if you can do this. Ming Hui has asked, what skills or courses would you recommend for scientific communications at the beginning of the field? So I would like to tell you that there are a lot of online courses available for basic scientific communication. Correct. Coursera has several courses. You can go through them. Editage, which is a body of Cactus, they also have a particular resource called Editage Insights, which publishes a lot of small articles about how to write something well. You can do that and refer to these, and definitely go and ask your university to conduct more such sessions by expert authors, right? People who have been associated with publishing industry, they can conduct such classes for you, and you will definitely learn more about scientific communication. Good question, Minghui. I really appreciate that. Okay, Sandhya says thank you. You are welcome. Minghui, you are also welcome. Bimla asking, like the American Medical Writers Association, is there an institute in India that offers certification or training in the medical writing field? I'm sorry, Bimla, I'm not very qualified in answering this question. I'm really not sure. Lokeshwar is asking, can you answer my question once more? What was his question? Is it always necessary to state the hypothesis or research question even when we state the objectives? So again, like I told you in the introduction, you are going to tell the larger goal. Say you want to say in the objective, you want to say that there is an unmet need, that a particular breast cancer doesn't have a particular treatment option available. So my clinical research aims to address that. So this is a larger goal that you are already stating in the introduction. But in your rationale or objectives of your method section, you are going to talk specifically about the clinical endpoints that you want to achieve. Correct? Say, for example, in the case of cancer, it is survival for so and so many years or so many months after the disease has been detected, or remission-free survival. Right? Some lot of terms are there. I'm not a very expert in the sense of cancer-related clinical research, but the objectives of the method section are tailor-made to the clinical endpoint that you want to achieve, and the overall hypothesis or overall objective is actually the developing the treatment for a particular disease. I hope I've answered your question. Okay, is there anything else? Shalini is asking, what qualities are the hiring committees looking for when they post jobs related to editorial or reviewer positions? I'm sorry, this question is out of scope for this session. Good evening. My question is how to write the introduction part in systematic review. Norin, again, the current session is about developing and writing protocols. For systematic review, there can be a dedicated session because it's a very different topic. Okay. If the research timeline has been completed, but the actual study or publication may be delayed, what kind of paper can be written in advance? I don't understand what you mean by a timeline has been completed. Maybe you're mentioning about the PhD duration, or you're mentioning about the timeline that you promised to your funding agency. I'm not really sure about what timeline you are expecting, but you cannot write something unless and until you have results, right? That's very straightforward. Maybe Dr. Indra, you want to come on stage and ask about this. Vasudha, is it possible to invite Dr. Indra on stage so that I can understand his question better? Okay. Never mind. Please connect with us later if you want to have more clarity on this. Okay, you are saying funded project, right? So you cannot publish unless and until you have findings. If your analysis is delayed for whatever reasons, and it has gone beyond the timeline that you committed to your grant agency, then you need to maybe go ahead and write to them and explain to them that these are the reasons because of which I could not complete this study, and the publication is going to take more time. And if things go well, maybe they will give you an extension. I'm giving you a very generic answer. This totally depends on the dynamics, the rules and regulations between your institution and the funding agency. Okay, we have 2 more minutes. Is there anything else that you may want to ask? Anything specific about today's session? Okay, then thank you everyone. Please use this link that Vasundhara has posted to fill the feedback, and do connect with us later if you wish to, and ask us your question. Tell us what other topics that you want to have sessions for, and definitely we will be conducting such similar sessions. Yes, we are ending the session. Thank you again.